Go right ahead if you want to talk about database-assisted MSMS deconvolution for metabolite ID, and I'll mute myself to let you go ahead.
Thanks, Tim. Let's get my screens oriented here. Let's see. Okay, you can see my slide. Can you see my mouse as well?
Yes, Gary, I can see your mouse.
Perfect, thanks. All right, well, thanks everybody. Welcome to day 2. Thanks for allowing me to be the lead-off hitter here. What I'm going to talk about today is 2 things. First, I'm going to talk about some of our recent computational efforts to accomplish MSMS deconvolution. And then I'm going to talk just briefly at the end about a metabolomics application that we're doing, looking at a relatively large cohort involved in the long life family study. Let's go ahead and start here with the MSMS deconvolution. And the reason that MSMS deconvolution is relevant pertains to chimeric MSMS data. So I thought that the best way to start this morning off was to give you a brief tutorial on what it is that I mean by chimeric MSMS data. I think most of us are probably familiar with this concept, but for those of us who aren't, I'll just give you a brief description here. So in the simplest situation, at least from an interpretation perspective, I'll depict that first. So what we have on the left there are 3 different molecules, 3 different compounds, and they're color coded. So you have a metabolite it has one M over Z colored in red, another metabolite colored in blue and colored in green, and they're all entering the mass spectrometer at the same time. But then we apply mass selection to isolate just the blue metabolite for fragmentation, and those thunderbolts there, those lightning bolts, are meant to depict fragmentation. And so what we then get is an MSMS spectrum that results just from the blue metabolite. And I say that this is the simplest of scenarios because all of the fragments that we detect are derived from the blue metabolite. And that allows us to match it easily to reference data. And those reference data are MSMS spectra. They could be from online databases, they could be from in-house libraries, but generally reference data in databases and libraries are generated from authentic standards. So you buy pure compounds from something like Millipore Sigma, or you have them synthesized, and then you fragment them and they're pure. And so we get a good match there, and this facilitates identification. What happens when we generate chimeric MSMS data is shown at the bottom of this screen. Here we still have our 3 different metabolites color-coded there, blue, red, and green, entering the mass spectrometer. But now instead of mass selection isolating just the blue precursor, we isolate the red and the blue precursor. And as a result, the MSMS spectra generated are said to be chimeric. That is to say that fragments are derived from both the red and the blue precursors. Now I've color coded those in the demonstration there on the bottom right-hand side of the slide, but of course in real life, they're all the same color and we can't tell which are derived from which. And this complicates things because the database spectra or the library spectra are pure and we don't get a match, and so that can limit our ability to do identifications. So we're most familiar with chimeric data, MSMS data, in the context of data-independent analysis. This has been a substantial focus in mass spectrometry. What I mean by data-independent analysis or data-independent acquisition is that you're not trying to get just MSMS data on one precursor, but you're making some sacrifices on mass selection to be able to generate MSMS data on larger numbers of compounds. You can do this by not actually doing any mass selection at all, just fragmenting everything that enters the mass spectrometer, or there are hybrid type methods such as swath that do impose some mass selection, but it does so with much promiscuity. So this is an example of a swath type experiment. What I'm showing you here at the top are the scans. So we start with an MS1 scan, and then you follow that MS1 scan with a series of MSMS scans. And the MSMS scans are done with some mass selection with 25 M over Z windows, so pretty large MSMS windows. And you can see that we use several of them in sequence to cover most of the small molecule range. So we cover from 100 to about 900 using all of these different isolation windows. And when we do that, we have to do it relatively fast. This is the time scale over which that occurs, and that represents one cycle. And the goal is to be able to complete these cycles. It's about a little over 3 seconds to do these cycles, and you do them over and over. You hope to get a couple cycles across a few cycles across a peak shape, so that you can get several MSMS data, but you're using relatively large isolation windows here of 25 m/z. And there are many variations of swath. Of course, this isn't the only way you can do it. There's ways in which you can do variable swath where the windows may change depending on what's present. A lot more innovative ways of doing it, but this is the general idea. So the question then becomes, how do we evaluate the amount of chimeric data that we're generating? So I've introduced here data independent acquisition, but how do we assess whether or not we're getting a lot of chimeric data? One way to assess the extent to which the data are chimeric is to use an approach that was inspired by this paper listed here in the bottom of the screen, an analytical chemistry paper in 2017 from Rick Dunn's group. And what you do in this approach is you look at an adjacent MS1 scan to your MSMS data, and you consider your targeted precursor, which is here colored in blue, and you ask how much of the intensity is coming from your targeted precursor compared to all of the other analytes present. So in this diagram, we would be asking what percentage of blue is contributing to this composite spectrum here. And that would represent the amount of red present would represent the percentage of contamination. So if you take this kind of approach and you apply it to the type of swath experiment that I explained earlier, so this is again a data independent acquisition experiment. And what I'm going to describe here are data from human plasma, where we did a 30-minute helix separation, so pretty standard separation. And we used 20 m/z isolation windows here. So the cartoon I showed you earlier was 25 m/z isolation windows, slight variation to that. And we consider how much of the data are chimeric. And this is the kind of picture that you get. So the y-axis in this diagram is the number of MSMS data, and the x-axis is the percentage that the data are contaminated. And as you can see pretty clearly, most of the data when we're doing this kind of DIA experiment, most of the data are chimeric. 98% of the data are chimeric. We have a small amount of data that fall into that first bin there, which are pure, but for the most part, not only are the data chimeric, but the percentage of contamination is pretty high. It's all up towards the 100%. You can see a lot of our spectra are in the 90% to 100% or above 85% contamination mark. So it's pretty expected, as I said. The fact that you get chimeric MSMS data in DIAs is something that people have thought quite extensively about. It's been an area of focus for many years in mass spectrometry. But what happens if we do a different kind of experiment, which is more common to do in metabolomics? The usage of DIA or swath-based methods in metabolomics they're not used as extensively in metabolomics as in, say, proteomics. But so what happens if we do an experiment that's a little more familiar to metabolomics, like data-dependent acquisition or DDA? So here we do the same kind of analysis using human plasma, 30-minute separations with HELIC, but instead of using a 20 m/z isolation window, now we use a 1 m/z isolation window. And what kind of chimeric data do we see there? So we still see, the important point here is that we still see a significant fraction of chimeric data. You can see that about 53% of the MSMS spectra still have some type of contamination from other precursor ions. So again, the y-axis here is the number of MSMS, and the x-axis is the percentage of contamination. You can see that about half of our data are pure, meaning that we're just looking at the precursor. But for those that we do have about half that are chimeric. Now you'll notice that the distribution of contamination is less than it is in the DIA data that I showed you, where we had a lot of the percentage contamination was up towards 100%. Here you see that it's more distributed, but there is in fact still a lot of contamination present. So you might ask whether or not this amount of contamination could influence our ability to make metabolite identifications for the reason that I showed you on a previous slide. Are we going to get chimeric data that doesn't match our pure library spectra, and will this inhibit our ability to make metabolite identifications? And this is a plot that I think communicates that it definitely can impede our ability to make identification. So when I'm plotting here on the y-axis is the percentage of contamination that's present, and I'm comparing that from unknowns and compounds that have been identified from the human plasma that was analyzed with that 30-minute helix separation using the DDA data. And what I mean here, for those of us that attended yesterday, what I mean here by unknowns is definition number C from yesterday's poll. So that is, these are features in the metabolomics data set that have not yet been identified. And what you can see is in those features that we haven't yet identified, there is more contamination compared to the features that we have identified, suggesting the possibility that if we could remove some of this contamination, or we could figure out how to deconvolute these chimeric MSMS data, or do a better job at deconvoluting these MSMS data, that it might allow us to make more identifications, even though we're still doing DDA experiments. So how can we go about trying to achieve deconvolution of chimeric MSMS data in metabolomics? I'm going to provide a couple of different solutions by which we can approach this problem. The first solution that I will describe is by using what I refer to here as sliding MSMS windows. So let me give you a diagram of what I mean by that. What I'm plotting here is an MSMS spectrum. This is a chimeric spectrum. We have the precursors on the right-hand side of the spectrum and the fragments on the left-hand side. You can see that there are two precursors, the blue precursor and the red precursor, and they're both generating fragments. That yellow arch there is meant to denote the MSMS isolation window. So you can see in this initial experiment, we are capturing both the blue precursor and the red precursor for MSMS analysis, and they're both generating fragments. But what would happen if we moved that MSMS isolation window slightly? When you're performing a mass spectrometry experiment, the user can set where that MSMS isolation window is centered. And so we have the ability to move it to the left or to the right. And you can see what happens because the MSMS isolation window isn't a perfect pulse, but rather has that Gaussian looking shape. When we move it slightly to the left, what we end up doing is capturing still a lot of the blue precursor, as you can see, but capturing much smaller amounts of the red precursor in the fragmentation experiment. And so as a result, all of the red fragments decrease. And we can do the same sort of experiment where we increase or we slide it to the right, the MSMS isolation window. And you can see now we capture much of the red precursor, but we're not capturing much of the blue precursor. And so you can slide the MSMS window around and you can look for fragments that go up or down in concordance with one another and use that to associate fragments to one particular precursor. And this is a solution that we wrote a software package to do this about 10 years ago called DecoMS2. And it works pretty well if you're using sliding MSMS windows. There is another way, however, to do deconvolution of MSMS data. And this is a solution that's been around. There's a long history of doing this, particularly in the GCMS world. Shusha Du probably has more expertise than any of us on this. It's something that's been done a lot with gas chromatography mass spectrometry data. And that is to use variable retention times between precursors to achieve the deconvolution. And the DecoMS2 software package that we wrote 10 years ago has the ability to do this. But we're also using a software package called MS Style that was published in Nature Methods in 2015 that came out of Oliver Fien's group. So let me just briefly highlight the principles that underlie our ability to do deconvolution on the basis of variable retention times. So again, I'm showing you a chimeric MSMS spectrum here. Fragments are on the left, precursors on the right, just simplified with that hash line to separate them. You can see both blue and red are orange. And even though the precursors, now I'm showing you an extracted ion chromatogram of the precursors, and even though the precursors nominally co-elute, you can see that there is a slight difference in when they come out. And therefore, if we look at these different fragments, the blue fragments and the orange fragments, they too should have slight differences in elution. So if you do a correlation comparison between the fragments and the precursors, we should see strong correlation between these product fragment pairs, precursor product pairs for the orange and the blue. And that's the basis in which deconvolution is accomplished with the DecoMS2 package and software packages such as MS Data. And there are other packages too that I don't have listed here, but those are the two that we've been using. So I'd like to just point out that these two solutions that I've highlighted thus far are really based on taking the experimental data and trying to deconvolute it. So I'm going to refer to those as front-end deconvolution because they do rely on taking advantage of the way in which the experiments are performed. There are some challenges, though, of doing front-end deconvolution. They do work very well, but they're not 100% effective. So they work often, but they're not always perfect. They're also highly dependent upon the workflow. So the success of these front-end deconvolution strategies depends on how the experiments were carried out. For example, if you are doing an experiment where you have poor chromatography and not very good separation, then trying to use chromatographic profiles to separate products and precursors is going to be more complicated. Or for example, if your precursors are really close to one another and you don't slide your windows in your MS isolation window the appropriate amounts, you may not get disparities between the different experiments that allow you to deconvolve. So it's very dependent upon the workflow. Furthermore, if we think about the environment where we are trying to share data, using public data, data sets, for example, that are in the Metabolomics workbench, they probably or most likely weren't performed with this kind of workflow, with sliding MSMS windows or getting a lot of MSMS data to try to do deconvolution across an entire chromatographic peak, for example. So there could be limitations, or maybe they don't have a good enough chromatographic separation to do these sorts of experiments. So there could be limitations in trying to extend these approaches to analyses where specific workflows weren't employed. So for that reason, over the last few years, we've been working on a computational strategy to perform back-end deconvolution. So this is in contrast to the front-end deconvolution that I've just described, now focusing on doing the separation, the deconvolution on the backside. And to do this, we've built a software package called DecoID, excuse me. And this is the architect of this is Ethan Stancliffe, who you heard from yesterday. So the idea, and I should point out that this is a solution, to the best of our knowledge, that hasn't really been implemented in metabolomics, but it was inspired by some different approaches that have been successfully used in the proteomics world. So the idea is that we have a experimental chimeric MSMS spectrum, but instead of trying to tease apart which components are contributed to the experimental spectrum, the ways that I've described on the front end, instead what we do is we take the reference MSMS data that is in our library and we convolve it together to try to recreate the experimental chimeric data. So we use different linear combinations of MSMS data from the library, from the MSMS library, and see if we can recapitulate the experimental data that we've observed. And when you can do that, you can get a sense for which precursors are contributing to the data. And to do this efficiently, we don't search every single MSMS spectra in the library, but rather focus on the spectra that are most likely to be possible contributors based on the precursor MLRZ value. So this is a picture of the DecoID interface. This was carried out in Python. So this is the TK anchor graphical user interface. There's a lot of different parameters here that you can pick from. I'm not going to go through all of these, but I'll just point out a couple of key points. One is you can select the library that you wish to use. As you can see here, we have an MZ Cloud option, a Mono option, a HMD option, but you can also use custom libraries. So if you have your own in-house library that you want to use with DecoID, that's perfectly compatible. I'll also point out that the success of doing back-end deconvolution relies upon having reference MSMS data. So when we start thinking about unknowns, this can be complicated because if there's an unknown that contributes to a chimeric spectrum, then it's going to limit our ability to deconvolute it with DecoID. So in that context, one of the approaches that we've been using is trying to build in reference MSMS data for unknowns, so that when you know that you have an unknown and you know that you're getting non-chimeric data, it's critical, obviously, that it's pure MSMS data, that you can store that in your library. And then in other experiments or other analyses where you might have chimeric data, you can use that pure MSMS data from the unknown to attempt to deconvolute. The other feature of DecoID that I would like to point out is the ability to use simulated M+1 isotopolog spectrum. And this one is a little less intuitive, so I'd like to take a moment to explain and diagram what I mean by that. So this is a picture of an experiment. Here what we have in blue is the target precursor. This is MS1 data. I should point out this is MS1 data. And the target precursor that we're trying to get MSMS data on is in blue. And what you can see is that the red compound is causing contamination, is causing the target MSMS data to be chimeric. However, it's not the red parent ion itself that's contributing to the experiment, the chimeric data, but rather it's the isotope of the parent compound. Okay, and the hash line there represents the MSMS isolation window. So this creates problems because our MSMS data are based on are generated from parent compounds, and we wouldn't have the isotope data in the database in our library to try to deconvolute this. And so what we did to try to overcome this occurs more frequently than we initially anticipated, where we refer to these as orphan isotopes because the parent isn't in the MSMS isolation window. And this occurs more commonly than we thought. And so what we did is we use a series type approach to predict the MSMS off the MSMS data that are in the databases. We use a series type approach to predict the fragmentation patterns for compounds that have a single C13 in this case. And that's proven to be pretty effective. So as an example that this strategy is working, I show you the data here. This is, again, we're looking at human plasma here. This is a DDA experiment, so a data-dependent acquisition experiment. We're using a 1 mol/z isolation window here. And what I'm plotting on the y-axis is the identification rate. So this is the number of features that we were able to successfully identify with high confidence as compounds. And what you can see here is when we are applying DecoID, our identification rate is higher than we're not using any deconvolution approach at all. Now you might wonder, you might say, well, that's an interesting approach to do the back-end deconvolution, but why sacrifice if you're doing the back-end deconvolution? I said that there's a long history of using the front-end deconvolution, so why sacrifice that? And indeed, a fourth approach to deconvoluting MSMS data is to use both the front-end deconvolution techniques that I've described in addition to the back-end techniques, the one that we've described here with DecoID. They're totally complementary. They're based on different principles and theories, so there's no reason that they both can't simultaneously be applied. And what I show you here is that, in fact, when we apply both of them, this is an experiment that is a DIA experiment. So this is a data-independent acquisition experiment, again, on human plasma, but here we're using 20 mol/z isolation windows. And what you can see here is that when our identification rate, when we combine MS-Dial and DecoID together, is higher than when we used either DecoID or MS-Dial independently. And perhaps not surprisingly, if we go in and look at the compounds that we were able to successfully identify with DecoID or MS-Dial, there are cases when they both worked, where they both successfully achieved deconvolution. But there are also chimeric cases where only DecoID worked and MS-Dial didn't, or only MS-Dial worked and DecoID didn't. So again, I think they're highly complementary, and I think using both in conjunction is a really good way to go. So in the last couple of minutes, what I'd like to do is say a few words about an application that we've been working on called the Long Life Family Study. And interestingly, this is an item that came up in the discussion that we just had previously, related to applying metabolomics to study healthy aging. This study, the Long Life Family Study, otherwise referred to as LLFS, is a huge multi-site, multi-investigator effort. It's sponsored, it's funded by the National Institute on Aging, but it has many universities, not only in the United States, but also in different countries involved. And essentially what we're doing is we're looking at families that are enriched with exceptional longevity. We're looking at 500 different families, all of whom are living long lives, not just the current generation, but prior generations. You repeatedly see these individuals living to be 100, over 100, 105, 110. I think we even have some that are over 110. So all of these families are enriched with exceptional longevity. The LLFS started in 2006, and they've been doing longitudinal blood draws on these participants. There are over 5,000 participants who are engaged participating in the Long Life Family Study, and they've been doing draws on them since 2006, as well as all kinds of other workups, imaging, and other types of clinical assays. On these participants, they're collecting a lot of different omics information. They're doing whole genome sequencing, RNA sequencing, proteomics, and we are contributing the metabolomics. And it's unique, at least for us, in that the amount of samples that we're analyzing is higher than the other experiments that we've performed in lab. So we're looking at about 15,000 different samples for metabolomics analysis. And I'll just point out that the PI, the leader of this study, is Mike Provence, who's at Washington University, a colleague of mine at Washington University in St. Louis. So how do we carry out these experiments? As I say, it's 15,000 different samples. And I should point out that we're analyzing blood. The way that we're doing this is we're using 96 well plates these are Captiva Agilent plates with solid phase extraction. And we're using acetonitrile and water to capture the polars. We're adding internal standards to those. We're using C13, N15 labeled amino acids. I wish we could add more internal standards, but unfortunately, some of the other mixes and standards that we wanted to add were approaching the $1 million cost. It was beyond the scope of what we could afford with this particular, beyond our budget for this particular study. Then we use ether and methanol to try to capture some of the non-polars. We analyze the polars using a 22-minute helic method. We analyze those in positive and negative mode using a 65-45 Agilent QTOF. We analyze the non-polars using a 31 method, HSS-T3, C18 Waters method, and positive mode on the 65-45 Agilent QTOF. I think there's certainly ways in which we can improve this workflow. As I said, we're trying to be conscious of budget limitations as well as time and some other factors, which is why we're doing it. By no means is this the perfect way to go, but it's been useful to us. The last slide that I'd like to share with you today has to do with some of the batch effects. I know that we're going to hear more about this, and there's been groups in the DTC who have focused specifically on batch effects. So I thought I would throw some of our experiences out and look forward to seeing others elaborate and say more about this as the day goes on. Obviously, when you run this number of samples, you do observe strong batch effects. This is a PCA plot. What I'm showing you here for each of our batches, because we're using 96 well plates, it's about 100 samples per batch. So I'm showing you 20 batches, it's about 2,000 samples. And batches that are earlier on are light, batches that are later are dark. So we go from light yellow to dark blue there. And you can see there's clearly batch effects. This is a quality control sample, which is a pooled sample that we made by taking small aliquots of the individual samples. And then we evaluated different, at least the approaches that are existing. We didn't have our hands on some of the stuff that the other DTC groups are doing, but we're very excited to hear about that. But some of the other normalization approaches that are out there. And what I'm plotting here on the x-axis is 14 different normalization techniques and normalization scores on the y-axis. That score is the ability to remove the variability from the quality controls, but retain the variability in the biological samples. And what we found is that the random forest technique performed the best, at least for our sample with these workflows. And when we apply it, that we're getting nice clustering of the data. This has proven to be useful. We're only about probably a third of the way through with these samples, but already we're seeing that SNPs that have been associated with having protective roles in healthy aging, protecting against pathologies such as Alzheimer's, are correlated with some of the different molecules and metabolites that we're seeing, particularly in the lipid space. So with that, I'll go ahead and wrap up, and I'd like to thank the individuals in the lab who contributed to these projects. As I said, the architect of DecoID is Ethan, and I've also pictured here the other individuals who've contributed to both DecoID and also the LLFS study. Igor Nikolsky was the architect of DecoMS2 back in about a decade ago, as well as the DTC group, and for all of the helpful and fruitful discussions over the years. And thanks for everyone for listening.